Hello guys, this is a presentation by Team 36 from the Chemical Engineering Department of KNUST. This presentation is by Daniela Apia, Christopher Ano, and Christodia Jamno. The Chemical Engineering Technology, Engineering Biology for a Sustainable Future. Table of contents include Introduction to Biochemical Engineering Technology, Biological Reactors, Enzyme Kinematics, Fermentation Processes, Downstream Processing, Case Study and Challenges, then the conclusion. Okay, so we are taking Introduction to Biochemical Engineering. Biochemical engineering involves applying engineering principles to biological systems and processes, such as fermentation, enzyme reaction, and bioreactor design to produce valuable products. It focuses on applying engineering principles to biological systems, including cell structures, enzymes, and microorganisms. This field plays a crucial role in advancing medicine, sustainable energy, and environmental remediation, making it a cornerstone of modern biotechnological innovation. One might ask, what's the importance of biochemical engineering? Biochemical engineering plays a crucial role in developing biopharmaceuticals, vaccines, and biofuels. It enables efficient production process for pharmaceuticals and biotechnology products. Advances in biochemical engineering enhances sustainability and productivity in these industries. Our first main topic for discussion is biological reactors. Biological reactors are specialized vessels where biological transformations occur under controlled conditions. These reactors harness the metabolic activities of organisms to produce biochemical products or degrade organic pollutants. Key aspects include optimizing parameters like temperature, pH, nutrient supply, and oxygenation to maximize the desired biological activity where we either want to produce pharmaceuticals, biofuels, or other products for industrial applications. They provide controlled environments for optimizing these reaction rates, substrate utilization, and product yield. Biological reactors are used by biotechnology, pharmaceuticals, wastewater treatment, and bioenergy production industries. Our next topic is enzyme kinematics. Enzymes are biological catalysts which accelerate chemical reactions within living organisms and industrial processes. Enzymes are protein in nature and they are synthesized by living organisms. Enzymes have active sites, which is where the substrate binds to, and enzymes are specific in their reactions. That is, they only work on specific substrates. Also, when an enzyme combines with a substrate, something known as an enzyme substrate complex is formed. And these are all structures of the enzyme that help facilitate its reaction. So how do enzymes facilitate reactions? One, by lowering activation energy. From the graph on your right, you could see that the initial activation energy for the reaction was higher, but due to enzyme activity, it lowered down into the blue graph, into the blue curve that you are seeing. So the low activation energy means that the molecules will need low energy in order to form products, which is a good thing. Then active sites. Due to the existence of active sites, enzymes facilitate specific reaction. That is, they do not just facilitate any reaction. So due to that, you can control the kind of reaction you are performing, and this is good too. Then we have an enzyme substrate interaction, which is what I've already explained using the lock and key model. So the enzyme is said to be a lock and the substrate is said to be the key. So when they combine, they form a lock and key complex. Roles in biochemical reaction. The roles enzyme play in biochemical reaction. 1. It enables regulation of metabolic pathways. 2. It facilitates diverse biochemical transformations in living organisms and industrial processes. Fermentation processes. Fermentation process is a metabolic process that produces changes in organic substrate through action of enzymes of microbial origin. In biochemical engineering technology, fermentation processes are extensively utilized for mass production of biochemicals, crucial in various industries including food and beverage, pharmaceuticals, and biofuels. Inoculation in fermentation, which is the first stage in fermentation, refers to the process of introducing a small amount of actively growing microorganisms, often referred to as inoculum or seed culture, into a larger volume of medium within a fermentation vessel, that's a bioreactor. This step is crucial in establishing a healthy and productive fermentation process. The next stage is the growth phase. The growth phase refers to a period during which microorganisms multiply and increase in biomass within a controlled environment, typically a bioreactor. The microbial growth occurs in certain phases. The first is the lag phase. This is the initial phase which occurs when microorganisms are introduced into a new environment. During the lag phase, cells adapt to new conditions, synthesize enzymes required for growth, and prepare for rapid cell division. The length of the lag phase varies depending on factors such as initial cell density, nutrient availability, and environmental conditions. The log or the exponential phase. In this phase, microorganisms experience exponential growth with the population doubling at a constant rate. Cells utilize nutrients efficiently, metabolize substrates for energy, and produce biomass and metabolites. The log phase is characterized by favorable environmental conditions such as adequate nutrients, optimal pH, temperature, and oxygen availability. The fermentation phase within the fermentation process specifically refers to the period during which microorganisms utilize substrates to produce desired products under controlled conditions. Microorganisms produce the desired product as a result of their metabolic activities. For example, yeast can ferment sugars into ethanol and carbon dioxide during alcoholic fermentation. Eventually, as these products are being formed, nutrient depletion occurs and accumulation of inhibitory byproducts lead to decline in microbial viability and metabolic fermentation. The decline phase marks the end of active fermentation when the desired product has been produced and harvested from the fermentation broth. Product recovery, which is the last stage in fermentation, in the fermentation process, involves the separation and purification of the desired biochemical products from the fermentation broth after microbial fermentation has occurred. So we've talked about fermentation. So what are some applications of fermentation in real life? It is used in the production of alcoholic beverages, dairy products, and fermented foods. It is also used in the synthesis of antibiotics, vaccines, and therapeutic proteins. Production of bioethanol and biodiesel as renewable energy sources is possible due to fermentation. Our next topic for discussion is downstream processes. Downstream processing refers to the steps involved in purifying and refining biological products after they have been produced by fermentation or cell culture in biotechnology or pharmaceutical industries. It is a crucial stage in the production of biomolecules such as proteins, antibodies, vaccines, and antibiotics. Some the stages in down processing. You take the first one, that is harvesting. Harvesting in downstream processing refers to the collection and separation of the desired product from biological source or fermentation broth. Downstream processing begins after cells or microorganisms have been cultured or fermented to produce the desired product. This can be enzymes, proteins, antibiotics, or other biochemicals. The first step in harvesting is separating the cells or biomass from the culture medium. This can be done through filtration, centrifugation, or sedimentation, depending on the size and properties of the cells. Cell disruption is a crucial step in downstream processing, particularly when extracting intracellular products, that is, products found in cells, such as enzymes, proteins, or other biomolecules. Cell disruption aims to break open cells and release their contents, which can include proteins or enzymes. This step is necessary because many valuable products are located inside cells and need to be extracted for further purification and use. So how is cell disruption done? We have mechanical methods by which you can do cell disruption. One is bead milling. In this method, cells are agitated with tiny beads in a chamber, causing them to rupture due to collision with the beads. Another one is high pressure homogenization. Cells are forced through a small orifice at high pressure, disrupting them by shear forces. And the final one is sonication, where we use ultrasonic waves, where ultrasonic waves create high frequency vibration that disrupt cell membranes. Okay. So I've talked about mechanical methods. So I'm going to talk about chemical or enzymatic method. Enzymes like lysozyme or cellulase can be used to break down cells or membranes selectively, releasing intracellular contents. This method is more gentle and can be specific but requires optimizing conditions such as pH and temperature. Detergents can also be used. An example of a detergent that is used for cell disruption is the Triton X100. You can also use alcohols or other solvents to disrupt membranes by affecting their structure. Catotropic agents like urea or guanidine disrupt protein structures and help release them from cells. Some physical methods can also be used. An example is the freeze-thaw cycles. This is where there is alternating freezing and thawing disrupt cell membranes by expanding and contracting water inside cells. You can also use electroporation, that is short electrical pulses which creates transient pulse in cell membranes, allowing intracellular components to leak out. So after cell disruption is done, the next step is filtration, where you remove large particles, cells, and debris from the solution. After filtration, chromatography can also be performed. At this stage, you separate components based on their affinity for specific materials. We have ultrafiltration and diafiltration as the next step. Ultrafiltration is primarily used for the separation and concentration of macromolecules based on their size and molecular weight. It allows for the removal of smaller molecules from solvents while keeping the larger molecules. Our next one is diafiltration. Diafiltration is a modification of ultrafiltration that involve the continuous addition of fresh solvents or buffer to retentate or to replace permeating components. It is used to improve product purity by removing small molecular weight impurities and exchanging buffer components. Formulation is the final step in downstream processing, and at this stage, the process of designing and optimizing the composition of a product to ensure its stability, efficacy, safety, and other desirable attributes are performed. It involves combining the purified active ingredients, such as the protein or enzyme you extracted, with appropriate excipients and additives to create a final product that meets specific requirements for storage, administration, and use. Some techniques that are used in downstream processing. One, filtration. This is when you separate solid particles from fluids. Two, centrifugation. This is where you separate components or or the solution you want to separate based on their densities. Then chromatography. This is where you separate the components of a solvent through, due to differences in their adsorption rates. Then precipitation. This is where you separate components due to due to the fact that they form solutes in certain solutions. Then finally, extraction. This is a way of separating two miscible liquids. Equipment used in downstream, we have bioreactors, centrifuges, filtration systems, chromatography columns, evaporators, and dryers. Major topic is case study and challenges. Examining complexity, a case study. So, so in this slide, you'll be taking a case study. You will take a problem, a real life problem, and try to see how biochemical engineers tackle challenges. Our problem was on the development of novel based therapy for cancer treatment. This problem would require genetically engineered cells for therapeutic protein synthesis. So the major challenges you would face will be cell culture optimization, that is achieving high quality cell densities and viability with suitable media composition, pH control, and nutrient supply. That will be a first challenge, of course. Then the next challenge you would face in trying to produce a genetically engineered cell for therapeutic protein synthesis would be protein purification. Obtaining high purity levels through efficient chromatography and filtration methods. Even though we have tools and equipment for these processes for purification, it's not all the time that they will be 100% pure. So getting this high level of purity is also a challenge that biochemical engineers are still trying to tackle in order to improve productivity. Then the last challenge will be to scale up. So transitioning from lab to commercial scale and regulating compliance and cost effectiveness. Sometimes it's easier to tackle a small challenge if you need to produce a drug for only one person. It's simpler than producing on a large scale. It's also because producing on a large scale, you also need to be effective. That is the time you consume and the number of chemicals and the waste management. All this will come into it. So you have to find solutions for all this. So this is why scaling up is a big challenge in biochemical engineering. This is our final slide, and we'll be concluding everything we've learned. So, in conclusion, biochemical engineering technology encompasses a vast array of disciplines and methodologies that are essential in shaping modern biotechnological advancements. From understanding biological reactors and enzyme kinetics to optimizing fermentation processes and downstream processing, each facet plays a crucial role in translating biological systems into valuable products. Throughout our exploration today, we have highlighted not only the complexity and intricacies involved, but also the transformative potential of these technologies in various industries, including pharmaceuticals, agriculture, and environmental sustainability.